So welcome everybody to the last session of this meeting. As Molly said, I'm Marilyn Albert. I'm at Johns Hopkins University. We've been hearing a lot about innovation in prior sessions, but this particular one is focused on innovative approaches in cognitive aging, which is meant to highlight some novel approaches that we might be able to incorporate into future studies of reserve and resilience. We have, as you know, 4 speakers on the program, but you'll notice that there's a 5th person at the table, and that's because this session, like 2 of the ones yesterday, is going to have an extra speaker who's just going to give a kind of snapshot of recent, very exciting data. So let me introduce the first speaker, which is Stephen Austad from the University of Alabama at Birmingham, and he's going to be talking about new and improved animal models for the study of cognitive aging.
Thank you. As preface, a number of years ago, I was invited to give a talk at a symposium on international strategic policies for nuclear deterrence, something which I know even less about than cognitive aging, but I did give that talk. To my horror, a couple of things I said showed up in an international report that came out of that meeting. This is just a long-winded way of saying that this is outside my normal bailiwick, but not as outside as some of the things. And what I say today, you should be informed by a couple of things in my background besides that. First of all, I've been studying the basic biology of aging for nearly 30 years now, and I just kind of eavesdrop on cognitive aging. But before I did that, I was a field biologist for a number of years, and before that, I was an animal trainer in the Hollywood movie business. And those animals, I would say, are better trained than any animals in your laboratory because they cost people millions of dollars if they don't perform. So what I'm going to do today is talk a little bit about animal models. This is the first meeting I think I've ever been at where people questioned the utility of animal models, and I think that's fully appropriate, but I'd like to just briefly mention what we can use them for and then suggest a sort of new approach to thinking about animal models. Why do we use, and I say non-human animals, because we are animals after all. In cognitive aging studies, you can do controlled experiments. You can do controlled experiments that are long-term in the life of the animal. You can look into cellular, molecular, and physiological mechanisms. Long-term studies can be done in a short time, and then they're inexpensive compared to the human studies that we've heard so much about. Now, having said that, I think there are certain things that we need to understand about animal studies. First of all, what you often hear, and I've heard it time and time again at this meeting, is that I worked on the mouse, the rat, the primate, the dog, whatever. My point here is going to be that there's no such thing as the dog, the mouse. There are only dogs, mice, primates, and rats. In animal studies, as in human studies, genetics matter. There is no thing such as the mouse. There are hundreds of inbred mouse strains. There are outbred mice stocks. There are wild mice. I'm just using mice here as an example. Despite that, about 80% of all the animal research that goes on with mice in aging is from one inbred strain and one sex in one inbred strain, although that's sounding. And there's a problem when we do that, is that we may be studying things that we really don't understand. So all of these inbred strains have their own idiosyncrasies, and some of them are known and some of them aren't. Here are a few of the idiosyncrasies of the C57 black 6 mouse, the most common one that we use, but it has a unique neurological phenotype that's only recently come to be known, and I think that's important. So let me just give you a cautionary tale. Study came out a couple of years ago to try to understand a rather obscure mouse disease that resulted in a widespread neuron loss. And it turned out it was identified as to be a result of this rather obscure receptor. But the interesting thing about that obscure receptor is that when that wasn't functioning correctly, you only got the phenotype in one mouse strain. And it wasn't even one mouse strain, it was one mouse strain from one origin, which the J with the underline there means it came from the Jackson Laboratory. It wasn't even seen in C57 mice from other sources. And it turned out that that strain from that source has a brain-specific transfer RNA that has a single nucleotide substitution that means that you only see this disease when you get a mutation in the single gene only in this one strain. So the take-home message from that is, could there be other neurological effects that we don't understand that are a consequence of that? We would have never known that if we hadn't looked at other strains. So not only do genes matter, but environment matters. And this is something that's well known to animal researchers, but maybe not to human researchers. These two graphs there, I'm well aware you cannot tell what's on the axes, but these are basically behavioral tests, motor dysfunctions that occur with the onset of Huntington's disease. And I think most of us think of Huntington's disease as pretty much the most purely genetic disease that one can think of. And you notice by 130 days of age, 100% of the animals get both of these neurological deficits, both of these behavioral deficits, in animals that were not enriched. The only difference between that and the lines in which never rise up above 20% in the one test and the other is that the animals were in what we call an enriched environment. But of course, what we call an enriched environment is not anything like the normal environment that animals evolved in. So all of our research animals, in my opinion, live in depauperate environments. What we call enrichment relative to what we would call an enrichment in humans is a trivial amount of enrichment, yet it has a huge effect, and we know about that. So from this part of the talk, the take-home messages are, there's no such thing as the mouse, the primate, the whatever. There are only individual ones. And that there are real advantages in terms of our understanding about any given species from looking across, looking at variation within the species and not just focusing on one single thing. That seems obvious to everyone here when thinking about humans, but it's not so obvious when people start talking about animal studies. And then finally, environment counts. So that's all a prelude to my real message here, which is that in the aging world, for the longest time, for those of us in basic research, what we focused on was how long animals lived. Can we make them live longer? And it was only when we became so successful at doing that that we started thinking about the fact, well, people don't want to necessarily live longer. What they want to do is they want to live healthy longer, or just healthy up to the bitter end. So really the field has shifted from a focus on lifespan to a focus on health span. And this is something that I've worked on quite a bit over the last few years. Health span, I think you can really break down into fundamentally three things: preservation of mobility, preservation of cognition, and preservation of sensory function. And when we were thinking about what animals could we do this the best in, could we really assess health span the best, it became quite clear to me that the best animal model for assessing this was the domestic dog, for a whole variety of reasons. And I'm going to spend the rest of my talk on the domestic dog because I think it's something that could be drawn into the cognitive aging field even more than it has been, and I'm aware that it has been to a certain extent, but I'm going to suggest something quite different. So what are the unique aspects of the domestic dog? First of all, you could argue it's the only animal species that's really adapted to interact fully with humans, and it's been selected genetically for that for thousands of years. It's been selected for a number of cognitive phenotypes, specific domains that are useful in performing certain tasks that people have. So some animals will hunt, some will track, some have been selected to be good at protecting or rescuing. They've been selected to be comfortable with and pay attention to and like to please people. And anybody who has a pet understands this. They're safe and they're easy to handle. So in my animal training career, I worked very briefly with chimpanzees, which are neither safe nor easy to handle. And the range of cognitive domains that can be assessed in these is absolutely astonishing because they can be trained to do many, many memory tasks. If any of you are familiar with service dogs, you'll know something about this. And they respond to vocal commands. And the thing that makes it really nice is over the last few years, the genomics of dogs and various dog breeds have really come to be developed. So I would now say that we understand and we know more about the genomics of dogs than any species, with two exceptions, that would be humans and mice. So all of these to me mean that the dogs are really poised to be useful in biomedical research in a way that they haven't been before. Now, what about the uses of dogs specifically for cognitive aging research? A lot of this has been done by Dr. Kottman and his colleagues over the years on beagles, research colonies of beagles, and it's been outstanding work, and I've been following it with a great deal of interest. But I'm going to suggest that there's some other things that we haven't really begun to explore what we can do in dogs yet, because like I say, I think any animals of our research animals have lived in depauperate environments compared to the environments in which they grew up. So here are some of the available opportunities that I think we could take advantage of. Some breeds cognitively age much faster or slower than others. We all know that small dog breeds live longer than large dog breeds, but it turns out they don't just live longer. They get cancer later, they get cognitive dysfunction later, they get cataracts later, they get all kinds of things. So there's this natural variation. Well, it's not natural variation, it's human-imposed variation for a great deal of variety in the way things age. We have selected for these specific cognitive domains in some breeds. So we could choose breeds that really focus on certain cognitive domains. Some of these may have less reserve. By the way, I thought I knew what cognitive reserve and resilience was before I came to this meeting. Now I'm not so sure. There are a great number of highly trained dogs that are available. And the advantage of highly trained dogs is they focus their attention incredibly on people. They're eager to please, and they're used to learning from humans. Let me just give you a few examples of what some of these dogs are capable of and what we could take advantage of in the cognitive aging game. They can stop or pause at designated locations. They can remember specific routes, open and close doors, turn lights on and off, find and retrieve specific objects, go pick up the blue puppet, put laundry into and out of washers and dryers like this one in this picture here, help dress and undress people. For you guys out there, they can be trained to lie still and get their brain imaged. And another thing is that service dogs that are highly trained to do all these tasks are retired at different ages depending on the breed, because of the differences in which they cognitively age. So retired service dogs might be an outstanding research resource. So we were asked to actually come up with a couple of recommendations or suggestions at the end of our talk. So here are mine. First of all, there's an ongoing study called the Dog Aging Study running at the University of Washington. The goal of that study is to enroll 10,000 dogs over the next few years in a long-term aging study, and this is a possible resource. So my recommendations in the last couple of minutes here is to develop, perhaps in collaboration with the Dog Aging Project, a registry of dog owners with specific types of dogs interested in using those dogs in research. And in fact, as listening over the course of the last 2 days, I think the dogs that belong to the subjects in some of your studies might be obvious candidates for this. The second thing is I've only had time to dip a toe into the use of dogs. I think the possibilities are enormous, and I think it might be worth planning a workshop to develop specific plans and protocols for the use of companion or service dogs in cognitive aging research. And I think the people that you want to invite to that should come from a broad range of expertise. And I think something very valuable could come out of it. Thank you.
Questions?
That's outstanding. I've said this before, and people have looked at me like I was crazy. So George Martin, who's at University of Washington, I mentioned to him this probably 5 or 6 years ago, and this was in terms of a Gates Foundation meeting. And you know, I said the exact same thing. There are millions of dogs that we have better control over their medical record history. They live in homes where people are completely engaged in their pets' welfare. The thing that you didn't mention, and I'll mention it, it sounds a bit crass, but almost all of these dogs go to death under controlled situations. So you would have access not only to brain tissue, but live brain tissue. You would also have control, and this sounds a little bit crass as well, to younger dogs that are put down in pounds, and you'd have live recovery of their brains. And for someone who does in vitro, ex vivo neurophysiology, I think this would be a great platform. And the things that we've learned in this meeting about the limitations of longitudinal studies, you could do this in dogs. You could do a longitudinal study and design the study so there wouldn't be savings. And that's a big problem for the humans, and it's almost impossible to do in rodents because they always have savings, because we ask them to do spatial tasks. Once they learn the cognitive map when they're young, they use it when they're old. Dogs are great at doing non-match to sample. You could have an unlimited non-savings related learning. So if you're looking for volunteers, count me in. Call me up. I'll be. You know, I think it requires people who do human work because they know how to do the clinical trials. It'll require veterinarians. So we have a great veterinary school up the road from me at Michigan State University. I'm sure we could get them engaged.
Thank you for making a number of the points that I didn't have time to make, but I absolutely agree with you. The fact that most dogs, when they do die, are often under controlled conditions means that these samples. My wife is a veterinarian. I actually have a canine metabolomics study underway now.
Yes, sir.
Steve, I should know the answer to this, but I don't. So in this big study of 10,000 dogs, I understand it would be primarily observational, not many interventions. How deep will be the testing for cognitive function? Will it be deep in all animals? Will there be subgroups? What's the plans for that?
You know, I don't really think, it's good. I don't think the plans have been formulated yet in that kind of detail. And it's one of the reasons I think that a planning meeting, would be a really useful thing, because we don't want to waste a big resource like this. We want to use it in the best possible way. And I think that the cognitive domain is a little bit different than we've been thinking about, I think, in terms of the dog study. Yeah, so this is why I think, So thank you.
Hi, thank you for telling us the advantages of using animal models. I don't question that there's definitely advantages of using animals for research, but the big question is really that if they don't share the same networks and same molecular pathways in humans, at the end of the day, whatever you find, you always have to check it in humans. And why do we still want to focus on, doing it in other animals, which have definitely certain species differences than humans? And can you really translate that to humans without actually checking it in humans again?
Yeah, well, that's a good point. And like I say, this is the kind of question I never get in other meetings, but it's a very good question. I would say that virtually every fundamental process of aging that we've discovered occurs in multiple species. Humans, after all, evolved from non-humans. We share many things. You notice I didn't talk about disease processes, because I think diseases are something that's highly idiosyncratic. And I'm kind of agnostic on whether we're going to learn anything about Alzheimer's disease from any animal models. It will be interesting to learn. But I think the fundamental process is what goes on in networks is going to be very similar across a lot of animals. I mean, just look at where did we discover adult neurogenesis? In birds, right? So there's certain fundamental things that are very much conserved across broad swaths of species. Now, dogs are reasonably close relatives in a zoological sense to us.
Yeah, I do know, I mean, I'd agree with you, there's a lot of similarities between the different species, but at the end of the day, isn't it better if we just go and find, using study those processes using human cells, human samples? I mean, like enrolling dogs is one way of studying the process, but we can also just enroll humans and study that process in humans. At the end of it, what do you get from doing the experiments in the dogs versus not doing it in humans?
Well, maybe we could talk about it more at the break.
Yeah.